All right, so let's first start about gene expression. So we went through how our genomes work, how they get duplicated, how they get repaired, etc. Right? But we still don't know how the genomes encode for all of the information in an animal body, in an organism's body. And that's, for me, at least, is world's greatest puzzle. Researchers like me and thousands around the world actually try to understand this, how the genomes encode for the information that end up building a whole animal like us, right? So one thing that we know for sure is that although different animals and organisms share the type of genes that they have, the end product is very different because in each organism or tissue, the kinds and amounts of the functional products that build particular cell types, ultimately the whole organism. In other words, the differences between our tissues and the differences between different animals is due to the types and the amounts of products that are encoded from the genome. Okay? So, for example, a good example of this is that in animals, the genome is the same in our muscle, in our neurons, etc., but the interpretation of the genomic information is different. The types of proteins that are in neural cells versus the muscle cells is different. And I don't know why, but this is on a timer, so let me see if I can get it out of there. All right. Okay. So how does that happen? There are two things that you need to know. When we talk about gene expression, we are talking about the process by which the information from a gene is used to make a product. Okay? When we talk about gene regulation, we're talking about the regulation of when, where, and how much a gene product is made from a gene. Okay? So, for example, gene expression is this process. You have the DNA sequence, that's your genomic information, that you're going to read the sequence and produce a functional product. In gene regulation, you're going to read the sequence and decide whether or not to make a product. So you can repress the gene and not make a product, or you can activate the gene and make the product. Okay? All right. The first step of gene expression, hence gene regulation, is the transcription of the DNA into RNA. And we call this the transcription. So the sequence on the DNA is copied into an RNA molecule. In prokaryotes and eukaryotes, the basic machinery that does the transcription is very similar. And here is a region of the genome where the transcription initiates. Okay? So you know that the RNA is synthesized from 5' to 3' by an RNA polymerase. There is a coordinate in the genome at the beginning of the gene where the transcription is going to start. And we call that, the coordinate we denote it as +1. Okay? +1 is the nucleotide where the transcription starts. Now, downstream, where the RNA polymerase is going to start transcribe of the gene is called are labeled with coordinates that are increasing from +1, such as +10, +20, +30 nucleotides. And this is called the downstream. And the upstream coordinates are labeled as you go negatively from -10, -20, and -30. As you go further, your negative numbers are going to increase, right? So here's the promoter region. So the promoter region is the region where it has the DNA sequences that are going to be important for localizing the RNA polymerase upstream of the gene in a way that the start of the transcription is going to start at +1. All right. I'm not going to go through this. You should know this already. How does RNA polymerase look like in prokaryotes? This is the E. coli RNA polymerase. It's a large enzyme complex, so that is made up of multiple subunits. Its protein complex is about 400 kilodalton or so. It has multiple subunits that do different functions. So this RNA polymerase complex is shown here. Here's the crystal structure, and the pink here shown is the DNA double-stranded DNA. And you can see how the RNA polymerase is sitting on the promoter from -10 to -30. You can see that it's making multiple contacts with the DNA through its multiple subunits. Now, the catalytic subunit of the RNA polymerase are called beta and beta prime. These both contribute to the catalytic, the core of the complex. And then you have the alpha subunits right here. There are 2 of them per the RNA polymerase complex. They're important for enzyme assembly, promoter recognition, and activator interaction. And you have the omega subunit here that is important for the enzyme stability. That is how the multiple subunits come together in a stable complex. Okay? All right, the stages of prokaryotic transcription, you have went through this already at the beginning of the semester, is the initiation, elongation, and termination. At the initiation stage, what you have is you have first the RNA polymerase binding to the promoter sequence with the duplex DNA. The DNA is still closed. It's not melted to open. So this is the catalytic center of the polymerase and the start side of the transcription, and this is the end of the transcription. The first thing that happens after the RNA polymerase binds to the promoter is that it opens the DNA by melts the DNA, duplex DNA, opens the 2 strands, and that we call a transcription bubble. Okay, and the RNA polymerase with this bubble is called an open complex. Then the RNA polymerase starts catalyzing phosphodiester linkage of the 2 initial RNTPs, forming this RNA, little RNA primer that it's going to add more nucleotides as it goes along, elongates and transcribes the RNA. All right. So the RNA polymerase enzymes that I just showed you is what we call the core enzyme. It can catalyze RNA synthesis, but it cannot find a promoter. So it cannot form a closed complex yet. Okay, so it's able to synthesize RNA, but it can't find the promoter on its own. The way that then where to start the transcription? If RNA polymerase just randomly started transcription all over the genome, that wouldn't make sense, right? So the proteins that help the RNA polymerase to start the transcription is called the basal or general transcription factors, and in bacteria, the example of these basal transcription factor is the sigma factor. So what sigma factor does is it recognizes sequences at the promoters of genes to recruit the core RNA polymerase complex to the promoters. Okay? So without the sigma factor, RNA polymerase can't find the promoters. So the the sigma factor plus the core enzyme, we call this holoenzyme, and that complex can find a promoter because sigma factor recognizes and binds to a consensus sequence at the -10 and -35 coordinates of the promoter. Now, this recognition is sequence specific, and we're going to show you the sequence in a minute. But the holoenzyme binds to from -50 to +50. That means it recognizes and binds to the DNA at these sequences, but not in a sequence-specific manner. Okay? But except the consensus sequence, in the consensus sequence at -10, -35, this additional binding is non-specific to the DNA sequence. What sigma factor does is it positions the RNA polymerase to start the transcription at +1 because of the way it recognizes the sequences at -35 and -10. So what are these sequences? Researchers took all the genes in the E. coli promoter and determined which ones are expressed the most. Okay, so and the genes that are expressed the most have strong promoters. Strong means it can promote a lot of transcription, right? And what they did is they know the transcription start site of each of these genes, promoter, so they lined them up. So they took the DNA sequence from the promoter region of each of the strong, highly expressed genes and lined them up at +1. And then what they did is they looked for the similarity of sequence, little sequence upstream of the transcription start site, and aligned that. So what they found is that -35 region of highly transcribed genes with strong promoters largely contains this sequence, TTG, ACAT, and the -10 region usually contains the sequence, TATAAT. Okay? And these two sequences are usually apart by about 15 to 17 base pair. Now, the promoters that is the most similar to these consensus sequences are the strong promoters, and they can support high levels of transcription. And promoters that deviate from these sequences at the consensus sequence, this consensus sequence at -35 and -10, are weaker promoters. We call them weak promoters, and they support less transcription. All right. So how do we actually find consensus sequences in the genome? This is done for a lot of things. We find consensus sequence for transcription factor binding sites as well. So how do we define consensus? So first we define consensus as something that U.S. Congress cannot achieve, right? But in molecular biology, we define it as the common sequence that's found at a set of regulatory sequences doing the same function, such as the promoter, right? So for example, we want to find the consensus sequence in this class of all the people, right? And I go around and ask everyone what they think about MCB, and let's say people have differing opinions, right? And these opinions will be basically represented as these letters. Let's say one of you guys said, I love MCPI, and then the other one said, I dope MCB1. Somebody said, I live MCB1. That's nice. The other one said, I know MCB1. I don't know what that means. Or I saw MCB1, etc. So what we do is to find the consensus of the class. We count the occurrences of each letter at each site and find the proportion. Okay? And then what we do is we represent that proportion of each letter at each side as the height of the letter. Okay, so for example, I is very conserved, right? It is at each side of these all of these responses that we got. L is less, right? It has a lower proportion, so we have a smaller nucleotide. O is a little bit more, and M is really at each of these responses, so we put a higher nucleotide, etc. There's going to be an example of the E. coli cap, I think, binding site at the NYU classes lectures where I post the lectures. So take that out and determine your own consensus from that. There's a study question at the end asking you to do it. And when you do it yourself, you're going to get the idea much better. Okay. All right. So in E. coli, there is not just one sigma factor. There are multiple sigma factors. And they can recognize and bind to different promoter sequences. And some of these sigma factors are specialized for specific functions. Okay? For example, here, the sigma 70 is the sigma factor that binds to the promoters of housekeeping genes that are highly expressed. Housekeeping genes are genes that are important for the life of the cell at all times. Give me an example of a housekeeping gene. What would be important for a cell all the time? Come on, guys. We just reviewed it like a few slides ago. What's important for a cell? Protein synthesis. We talked about RNA polymerase, right? That is something you always need, right? The gene encoding for the RNA polymerase would be a housekeeping gene, right? So these housekeeping genes, and especially the genes in exponentially replicating cells, these are genes that need to be transcribed at a high level. And the consensus sequence this sigma 70 recognizes is shown here. For example, here's another sigma factor called sigma 54. And this sigma factor binds to the promoters of genes that are important for nitrogen metabolism. And the consensus sequence that this sigma factor recognizes is different from the sigma 70. All right. So what happens after sigma factor binds to the promoter with the RNA polymerase? What it does is that it basically positions the RNA polymerase holoenzyme at the promoter so that the polymerase can start transcription at +1. After the transcription starts, the sigma factor is released. So while the rest of the enzyme complex moves along in the gene and starts transcribing, elongating along the gene, the sigma factor is released. And this is one feature that makes an initiation factor. That's a protein that's important for the transcription initiation, but doesn't get involved in the elongation. It gets released. There are hundreds of initiation factors in eukaryotic transcription, but one initiation factor in prokaryotic transcription is the sigma factor. All right. So after the sigma factor is released, the enzyme starts marching down the gene and elongates. And as it elongates the RNA, right? That's why we call it elongating. It's going to add RNTPs to make the RNA. So the polymerase advances 3' to 5' down the template strand, synthesizing the RNA in the 5' to 3' direction. It's going to melt the duplex DNA, so it's going to have this transcription bubble, and this bubble is going to move forward in the gene, adding the RNTPs and growing the RNA chain. All right, how does this actually look like? This is kind of looks like this. What you have is, here's a double-stranded DNA entering the polymerase from the left end and coming out from here, okay, to the right. So the polymerase is going in this direction, right? If it enters here and comes out from here, the polymerase is moving into this direction. What is striking is the polymerase is kind of like this closed structure, closing on the DNA, double-stranded DNA, right? You can't see the active site here. If you lift this a little bit up, 45 degrees this way, you can actually see what's going on inside. What is here is a transcription bubble, and this is the catalytic site that's adding the nucleotides to the growing chain of RNA that you can see here is a green molecule here. All right. So what we call that property is the RNA polymerase is clamping onto the DNA, right? It's basically literally holding the DNA. And that's the beauty about structure and function in molecular biology. The structure actually makes sense in terms of its function, right? So it's clamping the DNA tight. Here's a question for you. What would happen if the RNA polymerase didn't clamp tightly onto the DNA and it fell off? It's a pretty obvious answer. One of you guys can speak up. It'll fall off, right? Yeah. So what do we call that? What do we call that property of polymerases? Affinity is how strong it binds. It's related, but not quite. For polymerases, not falling off as it goes along. What do we call that? Processivity. Perfect. That it needs to be processivity. That means that an enzyme's ability to catalyze consecutive reactions without releasing its substrate, right? For polymerase, its ability to add nucleotides without falling off the DNA. All right. Give me an enzyme that has high processivity. Give me an enzyme that you know has a very high processivity. It does pretty good of a job. It can go for a long time. Count the DNA polymerases in your head. Which DNA polymerase or RNA polymerase would have high processivity? TAC, I'm not sure about, yeah, I think that's a really good example. What I was thinking is the DNA polymerase that synthesizes the leading strand, right? And I think TAC might be one of those. I just don't know if it's a leading or a, probably it's a leading strand synthesizing polymerase, right? Give me an enzyme that has crappy processivity. Well, I am not sure about that because the length of the Okazaki fragments are actually regulated. I don't think that's due to the lower processivity of the enzyme. It's due to some other things. Reverse transcriptase. We talked about how reverse transcriptase sometimes falls off trying to reverse transcribe the line RNA, right, in the transposons. All right. Now that the polymerase has a high processivity, it goes down all the way down to the gene and transcribes the whole gene. It needs to terminate the transcription. So the termination occurs at the transcription stop site. The polymerase releases the completed RNA and dissociates from the DNA. That's what we call termination. How does termination occur? In bacteria, there are two types of termination mechanisms. One is Rho-dependent and the other one is Rho-independent. Now, Rho is a protein complex that is a hexameric protein, I believe, that forms this interesting structure that is shown here. It forms kind of like an open ring structure. And what it has a helicase activity, and it disrupts the RNA-DNA complex with the RNA polymerase. Okay, how does this occur? So at the beginning, the RNA polymerase is elongating along the gene, transcribing the RNA. What happens is that some of the RNAs have a sequence that is recognized by the Rho hexamer. Okay? Once it recognizes that sequence, it binds to the RNA. And as the RNA polymerase moves, the Rho complex also starts moving. Okay? And it moves along the RNA, like just slides up. And if the RNA polymerase pauses at a certain site due to a special sequence in the RNA, we're going to talk about these sequences in a minute, that pauses the RNA polymerase, and Rho protein catches up with the RNA polymerase, and because it has a helicase activity that can unwind the RNA-DNA duplex here, right? Because the RNA is is duplex with the template DNA at the active site. It dissociates the RNA, and so that the RNA polymerase gets dissociated from the template, and the termination occurs. Let's say that you actually work in E. coli. You're interested in transcription, and you isolated a Rho mutant that slows down its migration along the RNA. What effect do you predict on transcription? Let's say you have this mutant, you isolated this mutant, and you looked at the transcripts in the E. coli that's mutant for this Rho protein, which makes it slower. What effect would you expect in these transcripts? They will have longer transcripts because they will not, if they do not catch up, they're not going to be able to catch up to the paused polymerase on time. And if the paused polymerase can pass that pausing, that is, if it's not pausing indefinitely, if it can go through, then the transcripts are going to be longer. Yeah?
Do you think mRNA processing afterwards is going to fix that?
RNA processing. Give me what do you mean RNA processing?
Like, what if you have a longer transcript and the excess that you have in its entirety, like, in contact?
We're in prokaryotes.
Oh, sorry.
Right? We don't have RNA processing in terms of splicing just yet. That's next lecture. All right. So when the RNA polymerase is actually paused, they pause at sequences that can form hairpin structures in the RNA. Those type of hairpin structures in the RNA actually destabilizes the enzyme sometimes if they're strong enough, or that cause the RNA polymerase enzyme to pause. Okay? So this is also the basis of how Rho-independent transcription termination occurs. What you have here is that you have, in those genes where there is row-independent termination, you have a set of DNA sequences where you have an enrichment of the C nucleotides and a G nucleotides. So these sequences that can, these are inverted repeats. Once these inverted repeats are transcribed into RNA, what they can form is these hairpin structures due to the complementarity between the inverted repeats. Okay? So this hairpin structure in the RNA that is right upstream of the transcription bubble pauses the RNA, or sometimes it actually terminates, destabilizes the RNA. Now, usually these hairpin structures by themselves is not enough to stop the transcription. What you have is usually these inverted repeats are followed by a stretch of A's in the template, and the interaction between the bonding between A and the U's are usually a little bit less than between the G's and C's, right? You know how DNA structure works. And sometimes this hairpin structure is going to be enough to destabilize the polymerase so that these bonds will be broken and the RNA will separate from the DNA template, terminating the transcription.
So the hairpin forms after the polymerase passes it. How does it end the transcription? Does that make sense?
So let's go back to the RNA structure, all the way down here. Oh, come on. Where is that? All right. So look here. What you can see here is that the active center, the catalytic center of the RNA polymerase is right at the transcription start site, right? But the RNA polymerase is making contact all over, right? And this is not the right picture. Also, the RNA is making contact coming out of here. So I think that we have a picture of that in the elongation. Yeah, this is probably a better picture. So now you can see that the transcribed RNA is coming over here, right? So this RNA is also making contacts with the protein. So this RNA, if it's kind of a nice and single-stranded, it doesn't affect the protein structure very much, right? But if it starts making hairpins right here, it starts bothering the polymerase. And that bothering is basically what destabilizes or pauses the polymerase, right? And that is due to, as soon as it transcribes, it doesn't kick it out. It's still complexed with it. Does that answer your question? Okay. Any other questions? All right. So that's transcription termination. Now that we went through the basically just the process itself, let's talk about regulation. In prokaryotes, gene regulation occurs in the genome in a peculiar way, in which that half of the genes are actually organized in approximately half the genes organized in operons, and they're co-transcribed, then translated into individual proteins, right? So these basically operons contain genes that are important for a certain process. For example, the trip operon has genes important for tryptophan synthesis. So the trip operon is composed of several genes, so that there is a single transcription start for all of these genes and a single transcription end site. And once the mRNA is made, the translation machinery has multiple start sites in this mRNA, leading to multiple proteins. Okay, so the regulation, this is what we call a coordinated regulation of gene expression. When there is a new, there is a need for tryptophan synthesis, you need to upregulate all the genes that are important for tryptophan synthesis. And one way to do that is put all of those genes into an operon and start the transcription initiation of all of these genes at once. Okay. All right. So this transcription regulation occurred through several proteins several proteins that are what we call transcription factors. These proteins bind to the gene regulator sites and activate or repress transcription through various mechanisms. So in the TRP operon, what we have here is a promoter, and it also has an operator sequence. Operator is a definition, a terminology that's specific to prokaryotes. So the operator is the DNA in bacteria that controls the transcription of an adjacent gene. And the promoter is defined as the DNA that directs RNA polymerase to bind and begin transcription. Okay? So in the tryptophan TRP operator, there's the promoter and operator sequences. And what do they do? Okay. At the start of the transcription site, upstream of it you have the promoter -35 and -10 sequences, and the operator is right in between. Now, this operator contains the DNA sequence that is important for the tryptophan repressor, the TRP repressor to bind. So this TRP repressor is a protein. In the presence of tryptophan, it binds to this tryptophan, which is a ligand, right? It's a small molecule. It binds to that ligand. And that binding makes the tryptophan repressor conformation such that it will be able to recognize these operator sequences and sit on them, okay? And that physical sitting, binding to the operator sequences, prevents the RNA polymerase to come in and bind to the promoter, okay? So it represses transcription by preventing RNA polymerase binding. Now, when the tryptophan is absent, that is, when you need the transcription of the TRP operon, what happens is that the repressor no longer has the ligand around to bind. When it doesn't bind to the tryptophan, it has a shape that is incapable of binding to the operator sequences, so it gets released from the operator. Now the RNA polymerase can bind to the operator and start transcription. Is that clear to everyone? All right, how does that happen molecularly? It happens like this. So here's the crystal structure of the TRP repressor. You have a TRP dimer, so the red one and the green one are two TRP repressors dimerizing, homodimerizing. And it has alpha helices that are going to sit on the major grooves of the DNA. And these alpha helices make interactions with the bases at the major groove, okay? And they will make stronger interactions with certain types of bases, right? So if its sequence is ATGC, let's say, if that's what TRP repressor is recognizing, it's going to make specific contacts with those bases. It also makes some contacts with the narrow groove of the DNA, minor groove of the DNA, also called the narrow groove. But these interactions are not sequence specific because they are going to be made through the sugar phosphate backbone. All right. So the TRP repressor binds to tryptophan, shown here, like this little molecule, binds to an allosteric site. That means a site that's not directly binding to the DNA, right? This site is not directly binding to the DNA. But it results in a conformation change in the repressor so that it can bind to DNA. So the tryptophan being here positions these helices so that they can sit into the major groove of the DNA and recognize the particular sequence. These are also called the recognition alpha helices because they interact with the bases in the major groove. And the repressor makes other interactions with the backbone of the DNA to stabilize its sitting on the DNA. All right. Which one of these interactions are sequence specific? The interaction with the bases in the major groove or the interactions in the backbone of the DNA? If you say major, raise your right hand. If you say backbone, raise your left hand. Excellent. Because the bases are in the major groove, of course, that's going to be sequence specific. Sugar phosphate backbone is everywhere, it's the same. All right, so that was TRP operon. Let's talk about now lag operon, which is basically the most famous example of bacterial gene regulation. the lac operon contains the enzymes that are important for breaking up lactose into galactose and glucose so that they can metabolize these as energy sources. So it makes the enzyme, beta-galactosidase, makes the transporter that's going to transport lactose, and transacetylase, which increases the specificity of the transporter for the lactose. All right. When do you need to start metabolizing lactose is when you don't have glucose, okay? And there is lactose around. Those are two conditions that are going to be required. So this is the promoter, the upstream sequences of just upstream of the lac operon. You have the promoter sequences, you have the operator, and you have what we call a cap site, and that is the site that's going to bind to the cap transcription activator. And the operator is the site that binds to the lac repressor. All right, and this is the transcription start site. So in conditions where you do not have any lactose and you have lots of glucose, you do not need to transcribe the lac operon genes, right? And the way that happens is that the lac repressor, in the absence of lactose, right, in the absence of the ligand this time, has the certain conformation that allows it to bind strongly to the operator sequence. And this sequence and binding prevents the sigma factor to be able to bind to the promoter. And if you don't have sigma factor binding to the promoter, you're not going to have the RNA polymerase binding to the promoter, right? And as a consequence of this, you're not going to have any mRNA transcription from the lac operator. If the E. coli is in a condition where there is some lactose and some glucose, E. coli, like us, always likes sugar. We like glucose a lot, right? And always prefers glucose. And what it does is it says, okay, there's a little bit of lactose. I can make use of this, but there's lots of glucose, so I do not need to transcribe the lac operator so much, right? And the way that happens is that in the presence of the lactose, the lac repressor binds to the lactose, and it has a certain conformation that prevents it to bind to the operator sequence strongly. So the lac repressor gets released from the operator sequences so that the RNA polymerase can bind and start transcription. But this transcription is at lower levels, so you have just a little bit of transcription. Because the lac operator, the lac promoter is a weak promoter. So I just said the lac operator is a weak promoter, so just the RNA polymerase coming in and transcribing gives you low levels of transcription. So do you expect lac promoter to deviate more or less from the consensus sequence at minus 35 and minus 10? If you say more, raise your right hand. If you say less, raise your left hand. More, right? Because it deviates from that consensus, so by itself it cannot support a lot of transcription. All right. Well, let's say we throw in our E. coli now in a condition where there is no longer any glucose, okay, and there's lots of lactose. Now, the E. coli wants to transcribe the lac operator at a high level, right, because it needs to metabolize the lactose. And how does that happen? So, in the absence of the glucose, there is a certain metabolite called cyclic AMP. And the level of the cyclic AMP goes high in the absence of glucose. Now, when cyclic AMP levels are high in the cytosol, this protein called the cap protein binds to the cyclic AMP, and the cyclic AMP induces a conformation for this cap protein that makes it able to bind to the cap binding site, okay, right here. So now the cap protein binds to its binding site. What it does is it makes connections, interactions with the RNA polymerase so that it stabilizes the RNA polymerase at the promoter and induces even more transcription. Right? In this case, cap protein is it an activator or a repressor? It's an activator, right? It's activating transcription, so you get higher transcription from a weak promoter. So that's the activator binding in the presence of high cyclic AMP. All right. So, in the lac operator, in the lac operon, right upstream of the operon, there are multiple operator sequences. So there is not only just one operator sequences that I showed you right at the promoter, there's upstream operator sequences. Okay? So what this is basically this. You have the regular operon here, right? So that's just downstream of the transcription start site. And then you have two more operators, which is called O2 and O3. One is much downstream of the transcription start site, so it's plus 412. And then one is upstream of the transcription start site, that's at minus 82. So what's the function of these operators? The function of these operators is to increase the local concentration of lac repressor in the vicinity of the promoter. Okay? So what happens is that you have a lac repressor. The lac repressor is a tetramer, so it's made up of two dimers. And there are only few of them. So there's only about 10 lac repressor tetramers per E. coli cell. If you have a few molecules, right, and the molecules are going to find their binding sites, and they're diffusing in the cell, right, all around, there's going to be a certain probability, a certain number of times where you're not going to have any lac repressor around to bind. And if that occurs, then the RNA polymerase might bind to the gene and start transcribing, right? And that's not very helpful because now you're transcribing lac operon in the absence of lactose, right? So how do you prevent this? The way to prevent this is not let the lac repressor to get away from the operator sequences too much, okay? And the way to do that is you can have multiple sites. Let's imagine that there's only one chair, right? And I'm free to roam around the stage, and I need to sit on the chair once in a while. And once in a while, I'm going to be over here and not sitting on the chair, right? But let's say there are 3 chairs, right? And I can sit on this chair. When I get up, there's another chair. I can sit over there. When I get up, I have equal probability of going anywhere. My chance of getting back in the chair is now much higher than my chance of sitting here if I was roaming freely around, right? Does that make it clear to you guys why the addition of operators can help increase the local concentration? Make sense, right? So actually, if you mutate these additional operators, the O2 and O3, you can reduce the amount of repression by 70-fold. So that increasing of the local concentration of the repressor through the other operators is actually very important for the very tight repression that you want to achieve. Okay, how does the lac repressor actually look like? It looks something like this. What you have is a dimer of the lac repressor, so red and the blue is a dimer, and it also forms a tetramer through tetramerization region here, making interactions. Now, the whole tetramer can bind to 2 different operators. So it can bind to one operator here, shown here, and the other dimer binds to the operator shown here, right? So if it releases this operator, it's still hanging on to the other operator, and so it's going to be still around the area of the promoter, right? Does that make sense? It's kind of like holding on with 2 hands. You're holding on one operator right here, the other operator here. So the lac repressor interaction with DNA is a dynamic process, so it's going to go on and off, right? And if it releases this, it might be still holding on here, so it's going to stay around the vicinity. So that's something specific to the tetramerizing lac repressor. Does that make sense? Any question about this? All right. So how does transcription, okay, we can understand how repressors can repress transcription, right? If it physically blocks RNA polymerase, of course polymerase is not going to sit on the promoter and start. How the activators work? The way that transcription activator works, it's a classic example of this is the cap protein itself. what it does is that at certain type of promoters, this cap protein makes physical interactions with RNA polymerase, mostly at the alpha subunit of the RNA polymerase. And those physical interactions with it is going to keep this RNA polymerase at the promoter longer and with more higher affinity, so that it is going to get a chance to make that bubble and start transcribing. Again, any molecule, any interaction in the cell, keep this in mind. These are biochemical processes, right? These are enzymes. The interaction of a protein with the DNA is going to depend on the affinity of that enzyme, the KD, dissociation value, right? And how much the concentration of the enzyme, etc. So at the initial steps of transcription, RNA polymerase II comes into the DNA. If it doesn't make the high affinity interactions and transition into an open complex, it can actually get off, right? So that initial interaction is also an on and off process, okay? The transcription factors, what they do is when the polymerase sits, it captures it, right? And it makes that interaction stable. Now that this polymerase is not going to be able to get off, so it's going to start transcription. This is one of the many different mechanisms how that they work, but this is the clear one that we understand as researchers. Okay, so let's learn some terminology because we need to learn how to speak molecular biology by the end of this semester, right? So when we're talking about gene regulation, we talk about negative regulation and positive regulation. So the negative regulation of transcription occurs through repressors. That is, a repressor binds to the DNA and keeps the gene off. Okay? A negative regulation of transcription can turn a gene on or off. And that depends on how the repressor works. Okay? So if the repressor, for example, if the addition of a ligand, for example, makes the repressor not able to bind to the operator, then that gene will be off. And in that case, if you remove the ligand, that repressor is going to bind. The gene will be off. And then if you add the ligand, the repressor is going to come off and the gene will be turned on. Does that make sense? Give me an example where this happens. We just learned about it. When the ligand is absent, it binds. When the ligand is present, it gets off. Lac repressor, right? When lactose is present, it's off. When lactose is absent, it binds and turns off the lac operon, right? So here's another alternative. For example, if the ligand is present, it might bind to the repressor, and that repressor binds to the operator and turns the gene off. And if you remove the ligand, then the repressor comes off and the gene turns on. Example of this. Trip, right? In the presence of the tryptophan, it doesn't need to make tryptophan, right? So the repressor binds. All right. The negative regulation can result in both activation and repression. It simply refers to the fact that the regulation is achieved through a repressor. Okay? So that's what negative regulation is. The positive regulation of transcription occurs through an activator. Okay? And the activator, for example, can also act differently in the presence or absence of the ligand. Let's say in this case, the activator in the absence of the ligand can bind to its binding sites and promote transcription. And if you add the ligand, the activator will get off its binding sites and the transcription will be turned on. Right? Does that make sense? So that's positive regulation of transcription, where there is a transcription activator regulating the transcription. Is trip operon positively or negatively regulated? What's the type of transcription regulation that occurs at the TRP promoter? Is it negative or positive? Negative, because it occurs through the TRP repressor. But of course, that promoter can be activated or repressed, right? That the negative regulation doesn't mean it's always repressed. It can be activated or repressed, but that occurs through the repressor. What about lac operon? Is it negatively or positively regulated? Both. The negative regulation is done by the lac repressor. What's the positive regulation done by? Cap protein, right? Because that's the transcription activator. All right, so that is the positive and negative regulation of the lac operon and the TRP operon. And in both of those cases, what we had is we had a single protein that responds to the presence or absence of a certain ligand, right? In the case of the TRP repressor, that was a protein that binds to tryptophan, and that binding determines whether it's going to bind or not to the DNA, right? In the lac, it was the lactose. In the cap, it was the cyclic AMP, right? So those are one-component systems. The signal is detected, and that determines whether the protein is going to bind or not. Now we're going to talk about a two-component transcription regulatory system in bacteria. All right. In two-component systems, an example of this is two proteins here that are important for the regulation of glutamine synthetase gene. So in the presence of high glutamine, what happens is you have this NTRB protein. The NTRB protein is made up of two domains. One we call a sensor domain, and the other one is a histidine kinase transmitter domain. This is called a sensor domain because it's going to sense the concentration of the glutamine in wherever it is. Okay? So if there is high levels of glutamine, what happens is that the glutamine binds to the sensor domain, and that binding results in a conformation of the NTRB protein that prevents the activity of this histidine kinase transmitter domain. Okay? So it binds and everything is closed. Now, the system has a second component, right? This is the first component, this is the second component, called the NTRC protein. The NTRC protein is also made up of two domains. One is called the regulatory domain, and the other one is DNA binding domain. Of course, DNA binding domain binds to DNA, that's why it's what it's called, and the regulatory domain regulates whether this DNA binding domain is going to bind or not. Okay? So that at high glutamine levels, you actually don't need to make a lot of glutamine synthetase gene, so the transcription is off. All right. What happens when you have low glutamine levels? So the glutamine is no longer binding to the sensor domain. So sensor domain releases the histidine kinase transmitter domain to be active. So the histidine kinase transmitter domain got its activity because it's a kinase. It phosphorylates the protein. What it does is it makes the phosphorylation in two steps. First, it phosphorylates itself at a histidine residue, okay? And then it takes that phosphorylation and transfers it to the aspartic acid in the regulatory domain of NTRC. That's why it's called a kinase and then transmitter. It transmits the phosphorylation, okay? So that regulatory domain, when it gets phosphorylated, it forms a conformation of the protein that allows the DNA binding domain now be able to bind to DNA, right? So the information coming from glutamine went through NTRB first and then relate to the DNA binding domain of NTRC. So that's how the two-component system occurs. Does that make sense? Questions? Okay. Here's a question for you guys. What's the advantage of the two-component system over a single-component system like the lac repressor or tryptophan, TRP repressor, or a cap? two-component system can relay the information from one protein to the other. What could be the advantage? Why would the cells might need this?
You have more amplification because each of the first component is multiples of the second component.
In this case, I wouldn't expect a lot of amplification because the actual phosphorylation is transmitted to another one. So the enzyme is not doing a whole bunch. It's like taking from itself and giving it, right? But there could be some amplification. That's certainly the case for eukaryotic signaling pathways, right? There's also another aspect of this.
Maybe multiple, like more regulation points, more points at which you can regulate the steps.
That's certainly. The first component tend to have transmembrane domains. That is that its sensor domain can be outside the cell, outside the cytosol, so that it can respond to extracellular signals as well, right? Because now that transmembrane protein, it can relay the information to another protein that can go and bind to DNA, right? The transmembrane protein itself is not going to be able to go and bind if it was a single component system. Does that make sense to everyone? Is that beautiful, right? You can have a system that can sense the cytosolic signal, and you say, I have a problem. I can't sense the extracellular signals. I'll come up with a two-component system. All right. How does the NTRC protein actually activate transcription? So what the NTRC protein does, once the DNA binding domain is able to bind to DNA, it binds to its binding sites that's upstream of the glutamine synthase A gene promoter. Now, this promoter is actually kind of special and is not very common in that this kind of mechanism is not very common in bacteria, in that the RNA polymerase, along with sigma 54, binds to the promoter but does not start transcription right away. What it does is it just sits there because it's not able to start transcription. When it is able to start transcription is that it makes a physical contact with the NTRC protein that is bound to the DNA upstream. And when that physical contact happens and you have this loop structure, the RNA polymerase now will be able to start transcription. Now that you can see that loop structure in the electron microscope, you can see the NTRC dimers here sitting on their DNA binding site. And you have the RNA polymerase with the sigma 54 sitting at the promoter. And once they can get together, the RNA polymerase can start transcription. So what NTRC does is it facilitates the transition between the initial binding of RNA polymerase to RNA. It's not POL2, sorry. It's just RNA polymerase, by the way. That was a mistake. Let me just fix that. POL2 is in eukaryotes. This is just RNA polymerase. In RNA polymerase, and the sigma to the formation of an initiation complex. And this requires ATP, which is unusual for bacterial transcription. All right. So because this NTRC binding site is quite far away from the promoter, and the binding of the fragment is quite far away from the promoter, and the DNA sequences at the NTRC binding site doesn't have to be in a certain direction. They can be in both directions. We call this a prokaryotic enhancer. So a prokaryotic enhancer is a short, like it could be less than 100 base pair of DNA that are bound by transcription factors, in this case NTRC, and it regulates transcription. It can be far away from the promoter and act in either direction. Can prokaryotic promoters act in either direction? So if you took the promoter sequence upstream of a gene, flipped it around, can it work? It will work to activate transcription, but will it activate in the right direction? No. Why? why does the enhancer work without direction? You can flip it over and it will work, but the promoter is not going to work. How does the promoter's structure regulate RNA pol II binding? What sequences are important? Minus 35 and minus 10. So these sequences are different, right? And the way that sigma factor sits on that is going to position the RNA polymerase. So, let's see here. So let's just go make a little cartoon. So you have minus 35 and you have minus 10. These two sequences are different, and sigma has a certain structure, right? Let's say that this guy is going to bind here and that guy is going to recognize here. And the surface of the sigma factor that's going to help the RNA polymerase position is going to position the polymerase in this direction, right? So if you flip this sequence then, how is sigma going to bind? It's still going to bind. This side of the sigma is going to recognize this sequence, and the other side that I drew very horribly is going to recognize this sequence. Now the interaction surface of that sigma is over here, and polymerase is going to position itself this way, right? So the direction of transcription is going to change because the sequences in the promoter are directional, right? That sequences are going to determine how polymerase is going to sit there. Now, if you flip an enhancer, because it's going to make a loop, it doesn't matter what direction the enhancer sequences is, the proteins that are binding to the enhancers are going to always be able to interact with the polymerase in a way that's going to support their function regardless of what their DNA binding interactions are. Does that make sense? Any question? Okay. We're going to talk a lot about eukaryotic enhancers. The concept is very similar. Okay? All right. So in the last 10 minutes, we're going to talk about a peculiarity in bacteria, which is an amazing mechanism that was discovered later, much later in bacterial transcription. That is that you can actually regulate transcription after the polymerase actually starts initiation. Okay? And this is called attenuation. The transcription is going to be stopped after it starts for a little bit. And the way that happens is, one of the examples of this is at the trip operon. So what you have here is, on the DNA, so this is the DNA, okay, you have this, and you have the rest of the genes that make up the trip operon. There is a sequence at the beginning of the DNA, upstream of the trip operon genes, called trip leader, trip L, okay? Now, this sequence is peculiar in that it actually can encode for an RNA that also has lots of trip codons, okay? So these are codons that the ribosome is going to need to make the protein. So it's going to be encoding for a lot of trip codons. In bacteria, transcription and translation occur at the same time, right? So what happens is if the trip operon the transcription gets activated, the polymerase comes in and makes this mRNA of the trip leader containing lots of trip codons, and as soon as the mRNA is made, the ribosome is going to sit onto this RNA and start translating it, making a peptide. Okay. When there's high levels of tryptophan, ribosome is not going to have any trouble finding enough tryptophan to incorporate into this peptide, right? And what's going to happen, it's going to continue just translating the RNA. And what that allows is that that allows is a special structure on the trip leader mRNA that forms a stem loop structure right close to the RNA polymerase with a stretch of use. What kind of termination does this remind you? Row independent termination, right? So what it does happen is that the stem loop structure that is very close to the RNA polymerase releases the RNA polymerase. So even though RNA polymerase started the transcription, with the high levels of tryptophan in the media, it's not going to continue go on and transcribe the rest of the operon. Now it's attenuate. Okay. What happens in low levels of tryptophan? What happens is that you have the same thing. The DNA, the RNA polymerase start transcription, making the leader. Okay. Now the ribosome also starts translating this trip leader and starts adding amino acids to the growing peptide. But this leader has lots of trip codons, right? But there is no tryptophan in the media. There is very low tryptophan. What will happen is that ribosome is not going to be able to find a lot of tryptophan to add to the growing chain, and it stalls. When it pauses at this region called 1, right, this pink region, what happens is that now the sequence in the regions 2 and 3 form a stem loop structure. And that stem loop structure is actually too far away from the RNA polymerase itself. And so it's not going to be able to affect the RNA polymerase stability. So the RNA polymerase is going to be able to continue its transcription. Okay? So the concept is this. If the ribosome is fast enough, it's going to cover both 1 and 2, so that 2 and 3 cannot make a hairpin. The hairpin that's made is going to be between 3 and 4. That's going to destabilize RNA polymerase. If the ribosome stalls, that gives a chance for the regions 2 and 3 to make a hairpin. And that will prevent the 3 making a hairpin with 4, right? And this hairpin is too far away from the RNA polymerase. It's not going to affect its stability, so the transcription can go on. Does that make sense? Question about attenuation.
How does it keep going if there's no tryptophan? Does it just not need any more tryptophan?
Oh, what will happen is this ribosome is going to get stuck there, and it's done, right? And it has no other function anymore, right?
And then another ribosome comes to the later.
Exactly, because remember, in the operon, the mRNA is long and it has multiple translation initiation sites, right? So another ribosome is going to come in, make the first gene. Another ribosome is going to come in, make the other gene, etc. Does that make sense? All right. So let's say that you want to adapt this attenuation. So what attenuation does, it makes the repression even stricter. What it is that if there was a chance for RNA polymerase to start the transcription in the presence of tryptophan, it's going to say, hey, wait a minute, you can't do it, so stop there, right? So it makes the repression even tighter. So this type of very strict repression is used, for example, in technology where people want to develop systems to produce a certain protein, etc., in bacteria, right? So what they can do is they can switch the transcription in the bacteria so that the protein is produced in only conditions that they want, right? So let's say that you actually want to adapt this attenuation and want to make it respond to histidine level instead of tryptophan level, right? So you want to trick the tryptophan. You want to use the same system to respond to histidine level. What would you need to change in the trip leader sequence in the DNA? What sequences you need to change? What do you need to do in trip leader to make the system respond to histidine instead of tryptophan? Change the trip codons to histidine codons, right? Because what will happen is in the absence of the histidine, RNA ribosome is going to stall at the histidine codons. Does that make sense? All right. So the transcription machinery and the basic steps of transcription is conserved between prokaryotes and eukaryotes. So here is the classic prokaryotic example, E. coli bacteria under a microscope. And here are you guys in a couple of years. Here's the Saccharomyces cerevisiae, which is the yeast that is a model eukaryote. So the RNA polymerases in Saccharomyces cerevisiae and humans are very similar and differ from prokaryotic RNA polymerases in one aspect. It is that in eukaryotes we have multiple types of RNA polymerases. We have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And the core enzymes that make up these polymerases are actually homologous to the bacterial enzyme, right? So you have RNA polymer RPB1, which is a homologue of beta prime, RPB2, homologue of beta. So these two make the catalytic center of the RNA polymerase 2 in eukaryotes, etc. And you have additional proteins that go along with the eukaryotic polymerases. They are bigger. The reason for that is that in eukaryotes you have different kinds of RNA polymerases, and they have adapted, they do function in transcribing different classes of genes. So in eukaryotes, RNA polymerase 1 transcribes genes that are encoding for ribosomal RNA. So it encodes for genes such as 20S, 18S, and 5.8S rRNAs. And so the function of this RNA polymerase 1 is in making ribosome components and protein synthesis. RNA polymerase 2 is the one that we're going to spend quite a long time next lecture. It transcribes just protein coding genes, most of the protein coding genes. So it transcribes mRNAs, some small nuclear RNAs, siRNAs, and microRNAs. So it has a role in all kinds of functions. And RNA polymerase 3 is specialized for another group of genes, which the most important one that you should remember are tRNAs. So it transcribes the genes that encodes the tRNAs. So the plants have actually more other additional RNA polymerases, 4 and 5, and they are specialized for transcribing a group of genes that make the pre-RNA for short interfering RNAs that you're going to learn in post-transcriptional regulation of gene expression. Okay, so see you on Wednesday.